is Brianne Giddings, and this is my Bio 201 project for Will Premack, Monday, Wednesday, Friday, 9:50 to 10:45, and I'm doing phototransduction in the rod cells of the retina. So photoreception is the detection of light, and the basic unit of light is called a photon. And so what happens is the light is going to go through your eye and it's going to hit the back of your retina. And in the back of your retina, you have two different things called rods and cones. And so today we're going to be talking about the rods because they detect light. So the basic structure of a rod is called, or you have a bunch of these individual discs. And then floating around all of these individual discs, you have something called cGMP, which is cyclic guanosine monophosphate, which we'll talk about in a second. And then you also have all of these sodium-gated channels. Now, one important thing to remember is that the rods do not like light. So the rods are turned off by light, but they're turned on by darkness. And so the main pigment that you're going to find in the rod is going to be called rhodopsin. And rhodopsin is split into two parts called opsin and retinal. And so retinal, we're just going to say that rhodopsin is going to look something like that. And then retinal is going to start off kind of squiggly looking. And in that form, it's called 11 cis retinal. Well, what happens is just like the rod, the retinal doesn't like light, and so when light hits the 11 cis retinal, it's going to cause it to change shape, and it's going to become more linear, and the 11 cis retinal is going to change into 11 trans retinal. Well, when the retinal changes shape, it's going to also cause the rhodopsin to change shape, and so rhodopsin is now going to look something more like that. And then you have, obviously, the retinal inside of it. Well, attached to the rhodopsin is a molecule called transducin, which is also known as the G protein. The transducin is an enzyme complex that binds with GTP and gets activated by hormones. Well, transducin is split into three parts called alpha, beta, and gamma. And what happens is when the rhodopsin changes shape, it's going to cause the alpha section to separate and go join up with something called PDE. Well, PDE is called phosphodiesterase, and that is an enzyme that breaks down cGMP. Well, like we said earlier, cGMP is floating all around the disc in the cell. And that cGMP is what's keeping all of these sodium-gated channels open. So when PDE attaches with cGMP, it's going to cause it to change over to normal GMP. When that happens, it's going to affect all of these sodium-gated channels, and they're going to start to close. When that happens, all of those positive sodium ions are not going to be able to enter the cell as well. And so the cell is going to hyperpolarize, and that means that it's going to turn more negative. When this happens, the cell is going to start to turn off and start to diminish, so it's going to kick signal off to a bipolar cell, which is attached to it, and then that bipolar cell will give signal to the brain, letting it know what's happening with the light. And that is phototransduction in the rod cells of the retina.